Well, hey, Living Environment fans, as of this moment, what you're watching this, we're more than halfway through the year of Living Environment. And think about all the stuff that we've learned about cellular respiration, photosynthesis, enzymes, and DNA replication, just to name a few. It's really been earthworms, it's really been brains. It's been quite a ride so far. Let's make the second half of the year just as good. So if you're watching this, you've already switched to a new classroom, and you're getting new seats in class today. And yeah, let's do the Bellringer. All right. Hey now. Mr. Kovacs class. It's Mr. Kovacs class. You might learn a ton. It's Mr. Kovacs class.
He's interested too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class. So last week our learning target was about how does DNA copy itself? And we're going to switch to why would DNA want to copy itself? And how does it make sure that the next cell gets the right copies? You'll see that that's, like in a bacteria, it's easy because bacteria usually has one strand of DNA. And so they copy that strand, and there's two strands. You put one in one new cell, one in the other. But in human cells, we have 46 strands of DNA that we have to copy. And then that's 92 strands sitting in one cell. And then you have to split those up so that the two new cells each get the same versions of the DNA, if that makes sense, right? It's like if DNA is a recipe or cookbook, right? You want each of the new cells to get the complete cookbook, not one to get pages 1 through 50 and then 1 through 50 again, and the other pages get 51 through 100, right? You want them to get the whole book, each one get the whole same. And so that's a little more difficult than you might think. That's what we're going to start studying. But let's review some things, should we? Replication. It's the copying of the DNA molecules, right? And it starts, right, in DNA, it starts with this replication bubble, sometimes we call it, right? Where you have helicase. Helicase splits the hydrogen bonds between A's, T's, C's, and G's. Ding, 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 ding. Right? And then polymerase comes in and works in basically one direction. So polymerase comes in and starts ding, 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 sticking A's to T's, C's to G's, that kind of thing. And it does it on both sides. And then when you're done, you have two strands. But this is what I find interesting too, right? There's the original strand was light blue. The new strands here were purple. Look at that, the two resulting strands each have half are light blue, the other half is purple. So one of the strands is always conserved, which means that it's possible that you may have sitting in your body right now a piece of the original DNA that first was ever copied by a living cell all 3.8 billion years ago. Mind blown. Okay, probably not because of mutations and things like that. But yeah. So yeah, when you copy your cells, each new cell gets one of the original copies. Yeah, one of the original ladder sides, and then the other side is the complement or the new one. All right, of course, why would a cell want to do this? Well, you copy it so that you can make new cells. And we're going to talk about the life cycle of cells this week. And our new learning target is how do cells, what are the important parts of a cell's life, I guess, is what our learning target is. All right? Cell cycle, we call it. One more thing. I may have mentioned this in class last week, but we're halfway through the year. I've already given 1,200 minutes worth of lab assignments. You need 1,200 minutes in order to be eligible for the Regents exam. I would say about 50% of my students, if it ended right now, would not be eligible because they're missing. Remember, you're missing lab assignments. You need to have the lab assignment is what counts. So sitting and doing the lab is great, but if you don't turn in the paperwork and it's not up to snuff, it's like you didn't do it at all in the eyes of New York State. So go back and check some of your lab reports. You should have them all in your classroom and or in your drive. And if there's any that are not up to snuff, fix them. Or don't, and risk not being able to sit for the test that you have to take at the end of the year, right? And having to try it again next August, and then next January, and then so on.